Hello, everyone. My name is Tian. I'm a research fellow from the Melbourne Neuropsychiatry Centre, the University of Melbourne. Thanks, Sarah and Mike, for inviting me to participate in this workshop on the topic of brain parcellations and functional territories. So in today's talk, I would like to touch base on the two models of brain topography. So one is brain parcellation, another one is the large-scale functional connectivity gradients. And I will discuss how can we use the two models to facilitate our better understanding of the brain organization. Okay, so I will start with the concepts of the two models, the parcellation and continuous gradients. I will then show how to map the functional connectivity gradients using functional MRI. And I would also like to show how to use the technique of gradientography to facilitate the null hypothesis testing and the model selection, and to enable parcellation of the Melbourne's parcellation of the subcortex. I will then discuss a few important validation steps once we have the parcellation generated, including the testing of the within parcel homogeneity, the replication of the atlas using a different imaging modality, and also the personalization of the brain atlas. So let's first look at the two complementary models of brain topography. On the left side, as you can see, that the brain organization has been studied for more than a century from the early 19th century, with mounting evidence suggests that the brain, the function and structure of the brain are topographically organized into distinct partitions with discrete boundaries. So this is the classic model of brain topography that has been studied for a long time and has been widely used in connectome mapping and other fields of neuroscience research. However, recent studies of cortical gradients propose that the topographic variation in brain structure, function, or connectivity might be better conceptualized in terms of a set of continuous gradients with gradual change in the absence of discrete boundaries. So this idea of connectivity gradients, the continuous gradients, somehow challenges the traditional notion of brain organization, and leads to the contention or debate about whether the brain is organized into discrete partitions or continuous gradients. So here are the two seemingly contradictory models of brain topography. However, are the two models really contradictory? So on one hand, we could continuously map the spatial variation in brain structure and function. However, we should also be able to delineate a hard boundary when the spatial variation is sufficiently large. So instead of asking which parcellation or continuous gradients, we ask how sharp a change along the spatial extent must be to warrant a hard boundary. So we conceptualize this question as model selection. I will discuss a bit more in later slides. But before I go into details of how to perform the model selection, I would like to show this slide, where this is anatomical location or simplified illustration of where the subcortical area is located. So unlike the cerebral cortex that has been studied for over a century, the subcortical area remains an uncharted territory for a long time, and which is highlighted in a recent review by Footman in 2016. This is partly because of the inherent imaging technique challenges in subcortical imaging. Also, unlike the cerebral cortex, where cortical parcellation generally covers the entire surface mesh, many subcortical structures are or have been studied in isolation based on anatomical landmarks without first establishing the functional boundaries between them. While only few existing connectivity-based or functional-based subcortical atlas that includes more than one subcortical structures, they generally do not preserve the anatomical boundary. So this leads the question of whether this discrepancy is because of the divergent structure and functional organization of the human subcortex, or it's because of a mass's limitation. So in the study, to address this question, we use the gradient mapping methods. And so we first continuously map the spatial variation in the functional connectivity across the spatial extent of the subcortex. And as I mentioned earlier, we will perform model selection, and we will only draw a boundary when the spatial variation is sufficiently large and justify the model selection. Otherwise, we would represent the topographic variation as a continuum. So just in case there are students who are new to the field of functional MRI, I quickly go through the basic concepts of fMRI and functional connectivity in this slide. So fMRI images were acquired in an MRI scanner that has 4 dimensions, which means so fMRI measures brain activation over time. So what we can use fMRI images, what we can do, we can measure local activation of a particular region, like region A or region B, or we can, from a larger scale, measure the functional coupling in the activation between the 2 regions. So the functional coupling is expressed as temporal correlation of the time series, and is what we call functional connectivity. So in order to map the connectivity gradients of the subcortex, we first need to compute the functional connectivity between each subcortical voxel and all the gray matter voxels in the brain. So this will give us a functional connectivity matrix. So each row in the matrix represents a whole brain connectivity profile for each subcortical voxel. And so we can then quantify the similarity in the whole brain connectivity pattern between each pair of subcortical voxels. So then a traditional way to do the parcellation is to apply the clustering approach to group voxels into clusters. So each cluster then represents a distinctive subregion or region that comprising voxels share similar whole brain connectivity pattern. However, this method, whatever the clustering algorithm you want to use, this method assumes the existence of discrete partitions, and it is often a challenge to determine the optimal number of clusters, which is another story to tell. But here we don't use the clustering approach. What we did, we used the Laplacian eigenmaps, the method called Laplacian eigenmaps, and decompose the high dimensional similarity matrix into a few low dimensional connectivity gradients. So after the eigen decomposition, each eigenvector represents a dimension of connectivity differences across topography of the subcortex. So to simplify this talk, I will only focus on the first principal gradient as it explains the most variance. So here is when we project the value for each subcortical voxel onto the brain, we have this eigenmap. So here is the gradient map. So by definition, eigenvector has 2 properties. One is the direction, another property is the gradient. So because of that, we first made the gradient orientation or the gradient direction for each subcortical voxel in this gradient field, and then we fit tensor into the gradient field, such that the shape along each tensor just indicates the gradient direction, whereas the length of the tensor represents the gradient magnitude. In this way, what we can do is to propagate millions of string lines in the tensor field in the same way as white matter tractography is typically performed in diffusion imaging. But here we proposed a tractography that is based on connectivity gradients. So we simply gave it a name as gradientography. So let's look at this picture. Each string line represents a trajectory or maximal change in the eigenmap. And here the string lines are colored by the gradient magnitude in each voxel. So we can see that locations with like red location or here with those circumscribed band indicates the local maxima in the gradient magnitude, which then suggests the putative functional boundaries. So in this way, the gradientography enables not only a striking visualization of the brain gradients, but also the detection of gradient magnitude peaks. So let's take a closer look. Here are the string lines that travel through the left and right, through the dorsal part of the subcortex, traveling through the globus pallidus, putamen, nucleus accumbens, and the caudate. So what we do next is to project the three-dimensional gradient magnitude to a two-dimensional diversity curve. And we can clearly see that, say, as three peaks corresponding to the locations of the functional boundaries. Similarly, we do the same approach for the ventral part of the subcortex, where we see string line propagating through left and right thalamus, hippocampus, and amygdala. Again, we see a lot more local peaks or local maximum presented in the diversity curves. So now the question is, shall we consider all the local peaks? What if those peaks are purely just driven by chance, or other confounds are driven by the effect like spatial smoothing and geometric contraction that is quite unique to the subcortical structure? So here, to control those factors, we say, okay, we need to generate a null model to benchmark those gradient magnitude. And here we generated such a null model that shown in gray. So we only parcellate the subcortex at locations when the empirical peaks are significantly larger than the peaks in the null diversity curves, such as the null can be rejected. Otherwise, we argue that the spatial variation in functional connectivity is best represented as a continuum in the absence of discrete boundaries. So given that this is educational workshop, I just want to talk a bit more about the null model. So here are two axial slices for the caudate, putamen, and the globus pallidus, where the background image is the gradient magnitude. So the slice shown on the left is the graph computed from the observed fMRI data, while the one shown on the right is the revised graph represent the null. So what we can see that there's nearly no edges. Also here the edges or the edges of which is the red lines. They represent, so they are drawn between subcortical voxels that share similar connectivity pattern. And while we can see that there's nearly no edges traversing across the boundaries, crossing boundary between putamen and the globus pallidus, whereas in the null, we see a number of rewired edges that traveling through the boundaries. So this means that, so this is consistent. So like here, we did not see a gradient magnitude peak in the null, but we see a clear gradient magnitude peak in the observed data. So this suggests that the null hypothesis can be rejected for this particular boundary. So here is just an example, a hypothetical example to show how the effect of geometry or geometric contraction in the subcortex can lead to a superior boundary delineation. And so here we see the edges. So here in this graph, those edges are just randomly placed along between each pair of voxels or pixels more precisely. And we see that, so those random placed edges across especially this place, across the bow tie area bow tie is consistent with a uniformly distributed gradient magnitude, a uniform spatial gradient, which means that the rate of change in the gradient is homogeneous across the extent of the bow tie. While we see that, and this eigenmap is computed from the graph, so does the gradient magnitude. While we say that because this is a uniformly the homogeneous gradient magnitude, so the spatial gradient is by definition homogeneous. So the null hypothesis is true by definition, but we still see a gradient magnitude peak presented in the gradient magnitude image, suggesting providing evidence of putative boundary. So, but we know this is not true. So if we don't consider the effect of geometry, we will end up delineating more functional boundary just simply because of the geometric contraction. Okay, what I already mentioned about some of the more important steps in the model selection, and now so we use the model selection and then now hypothesis testing, we generated the initial parcellation for the human subcortex that have 8 bilateral regions, so 16 in total. So as you can see that they correspond quite well with those anatomical boundaries. We next map the connectivity gradients for each of the 16 regions, and with the performing of model selection recursively, this yielded a hierarchical parcellation for the subcortex that extends to scale 4. We could not further parcellate subcortex because the null cannot be rejected. However, I would like to note that not all subcortical structure has 4 scales of organization. For example, the globus pallidus only extends to scale 2, comprising two subregions, whereas the thalamus and the hippocampus, they has more hierarchical scales. We think this is consistent with the functional and organizational complexity within the thalamus and the hippocampus compared to the globus pallidus, where the globus pallidus they has more specific and unique or simple role in the regulation of motor control. But we know that hippocampus and thalamus, they have so many different roles in like memory, in learning, and spatial learning, and emotional learning, emotion, etc. So okay, we have generated the parcellation. As I mentioned at the very beginning, it is important to validate your parcellation. So one of the most fundamental thing to validate is the within parcel homogeneity, because that's the fundamental principle of any brain parcellation. So what we did here is I want to mention that because the parcellation was generated based on functional connectivity. So to avoid circular analysis, when we compute the homogeneity, we compute the homogeneity in terms of the fMRI signals within each region, not at the connectivity level. And also we use a different data session. So here we do the same thing for the actual parcellation and a set of random parcellations. We also compare the homogeneity of our parcellation to several existing parcellations. What we find that in general, our parcellation outperform the random parcellations and also several existing parcellations, including those for the hippocampus, thalamus, striatum, and also 2 atlases for the entire subcortex. The next validation steps is to validate parcellation using different imaging modality. Here, I just show one example of replication using 70 fMRI. So we can see that they all look similar. It's just an example of scale 1 parcellation. They all look quite similar, and 70, the boundary even sharper, maybe benefited from the better spatial resolution and also the contrast to noise ratio. By comparing the atlas delay generated from each scale, we show that they are quite similar, or they have spatially quite the spatial correspondence is quite good and significantly outperform the comparison to random parcellations. The final step of validation I want to show is that is to show whether the group consensus atlas can represent individual connectivity architecture. This is of high practical importance, and because we know that in some occasions, so the if the group parcellation cannot represent individual, then the individual specific atlas will be beneficial, maybe can result in improved treatment for those targeted clinical practice, for example, the brain stimulation. So in this case, to address this question, we use the SVM classifier, like we're using a machine learning approach to derive personalized subcortical parcellation. In briefly speaking, here just a very simple schematic representation of the methods. So what we did, we trained a binary SVM classifier to classify whether a voxel, that like the feature, it belong to a region or it's belong to the surrounding area. So once we have the model trained, the classifier trained, we apply the classifier to a new subject, and use the classifier to predict an unknown voxel to predict the posterior probability of this voxel belong to the region of interest or the region that is outside. So by doing that, we generated a personalized parcellation for each scale 4 atlas. And here just an example of the personalized atom atlas in 2 subjects. So we use dice coefficient to quantify the similarity in the atlas between individual specific to the group atlas. Well, we can see that this subject show better dice coefficient, this one show lower dice coefficient, and that the boundary between different regions they are evident. Clearly, you can see those variations from this picture. And here, just to show an example of the distribution of dice coefficient across individuals, across all the testing individuals, for 2 regions. One is the dorsal posterior putamen, another one is the hippocampal body. As we can see that the dice coefficient is quite high, about 0.8 for most of the subjects, which means that the group consensus atlas is representative of individual, at least for those subjects. However, we also see a few proportion of individuals, their connectivity architecture might differ a lot from the group that reside at the tail of the distribution. However, so those subjects may relate to some unique or deviation in the anatomy, or because some other factors in the study we did not look into further, but it will be something interesting to address in future work. So as a summary, or the things I want to express through the workshop is that, so yes, there are 2 complementary topographic models of brain organization, the parcellation and continuous gradients, but they are not necessarily contradictory. So here we could, by model selection, we could provide a reconcilable, way we can reconcile these 2 models of brain organization. And also I want to say that it is important to use statistic non-hypothesis testing and model selection to avoid superior boundary delineation, to avoid overparcellation of the human brain. And a few other important remarks, even though I did not cover all of them, but I think there are just some general principles that will be important for brain parcellation studies. So first, it will be important to validate brain parcellation if you can, using different data sessions. And also, if the brain parcellation is generated from one modality, for example, fMRI, it will be good to compare that with anatomical and histological maps, or some other imaging modalities. Or if the brain atlas is derived from resting state, it will be very interesting to see how they perform in task fMRI. And if possible, would be beneficial, would be useful and to validate your brain parcellation using a different data sets to control like the demographic details and age range. Also, it's a way to address, it's also a way to address the generalizability. Okay, that's everything I want to talk about in today's workshop. And I would like to thank my supervisors, my supervisor Andrew Zalaski, and also other collaborators, including Michael Brixby and Daniel Margulis, for their contribution to this work. Most of the things I talk about can be found from this paper, and the atlas is available to download from the GitHub page, and also those May scripts or the MATLAB codes to generate and to run some of the now hypothesis testing, and also the gradient mapping can be, those scripts can be found on the website. And I hope you learned something from today's talk, and happy to take questions.